Hello, everybody. Welcome to our second episode of our webinar series. Today we will have a joint webinar with Cytosurge from Zurich. And with me today is Paul Monnier from Cytosurge. My name is Martin Winter. I'm the founder and managing director of Lab Automation Network. And Cytosurge is one of our network member companies offering outstanding technology for single cell injection, single cell handling, and other quite exciting applications, which Paul will show you today in the webinar. Let me just introduce you to a couple of informations. This webinar will be recorded so that we can publish it and give other people the opportunity to watch it later on. And of course, it's possible to ask questions. We will have a Q&A session at the end, but you can, if you click on the Q&A button in Zoom, you can pose your question at any time, and we will even try to answer it during the webinar or in the Q&A session, or if there are too many questions, even after the session via email. Again, thanks for joining. We are starting now with the webinar. The webinar series is about outstanding technologies. And we have made a selection of those technologies for this year. More webinars are coming after the summer holidays. We've already had our first episode 2 weeks ago. Later this year, we will have further episodes on digital liquids, completely new methods for analytical technologies, robotics and automation, and also on special technologies to capture processes, laboratory protocols with semantic annotations, and much more. But today, again, we will have our webinar on the FluidFM technology, which is a combination of atomic force microscopy and nanofluidics. Paul will introduce you into that, and what you can do in particular in the field of neuroscience applications. So Paul is from Cytosurge AG in Zurich. He's a molecular biologist, and he's working with Cytosurge for a couple of years now, coming from the ETH and the Pierre and Marie Curie University in Paris. And I would say he's the expert in FluidFM applications, and he's leading the biolab at Cytosurge in Zurich. I have introduced myself already. My name is Martin Winter. I'm a chemist by training, and I founded the Lab Automation Network 12 years ago. So just as an introduction to our organization, the Lab Automation Network is an alliance of companies that are offering solutions in the field of lab automation and digitization. As I said, Cytosurge is one of those companies, and we are covering a wide range of applications in labs, in pharma, biotech mainly, but also chemistry, cosmetics, and food industry. And we are offering, together with our network partners, technologies that range from robotics and automation, protocol design for laboratory protocols, liquid handling, microfluidics, and many, many more, including software solutions and digital technologies for the digital lab of the future. And we're also offering consulting services, for example, for requirements engineering. So if you plan to introduce new technologies to your lab, don't hesitate, come to us, and we're happy to assist you. Okay, but now I'd like to hand over to Paul Monnier from Zurich for his talk on FluidFM and applications in neuroscience. Thanks for joining us, Paul, and the stage is yours.
Yeah, thanks a lot, Martin. Thanks for this nice introduction and for inviting Cytosurge and myself to be part of this webinar series on outstanding technology. I cannot disagree on this fact that FluidFM indeed offers really unprecedented application. It enables things in life sciences that are not possible without FluidFM, and we will see that today more particularly in the field of neuroscience. Let me, oh, cannot switch the slides. Yeah, okay, it's slow. So today, the webinar will first take a few minutes to introduce you FluidFM generally. The timing here are approximative, but let's say 10 minutes on the introduction before really diving into the application in neurosciences. We will see.
So Paul, I cannot hear you. Maybe you have an issue with your audio. Is that possible? No, we cannot hear you. Maybe you try to.
Better now?
Now we can hear you again.
Okay, it's weird. It was working just before. I was hearing you very well. Okay, so did you hear anything or not at all?
Yeah, it stopped quite at the beginning when you shared your agenda slide. Okay, so yeah, let me just restart then. So I will briefly first introduce you approximately during 10 minutes FluidFM, generally the technology, what it is, before really diving into neurosciences applications. We will see there is a broad range of application, but I will try to give you nice insight into what is possible with FluidFM, with some published data, but also some unpublished data that have been obtained either by us at the research or by some of our users around the world. And as Martin mentioned, there will be a Q&A session at the end, so don't hesitate to ask questions via the Q&A tool here in Zoom. Okay. Okay, there is some lag. Okay. So first, what is FluidFM? FluidFM stands for fluidic force microscopy. Why? Because it is a combination between microfluidics system and atomic force microscopy. So basically, we use this atomic force microscopy probe here that are hollowed with a channel inside that you can fill with liquid that you can then push through the probe by applying some pressures. And there is this atomic force microscopy component, which is this laser feedback, this force control feedback, which is a laser reflected on top of the probe that we will see allows to really control the position of these probes, to know where it is, or when it touches a surface, it will bend the probe, and the force that is applied on the probe can be precisely measured. An important component of the technology is this microchannel probes. We can see here on this series of pictures how it looks like in real life. So you have really the probes glued on these plastic adapters that are made to be easy to handle, with a 1 microliter reservoir here that you can fill in with any liquid that you want. We will see later, for example, to inject compound into cells, for example. Here, if you look a bit more close, you can see that these probes are really, really tiny, like 30 micrometer wide, and they have at the end here, for example, in the case of the nanosyringe, an aperture in pyramid that we, for example, use for injection into the cells. We have different, it exists different sort of FluidFM probes. Some flat probe, the FluidFM micropipette, where you can have different aperture size that you can use to, we will see later, to isolate cells, for example. The nanosyringe with a pyramidal shape that we can use, we will see later on, to inject into cells, or what we call the FluidFM nanopipette, which is very similar to the nanosyringe, except that the aperture of the tip is located really at the apex of the pyramid. From this different shape, as I already briefly mentioned, we can
a lot different, what we call FluidFM mode, FluidFM application, or yeah, we call them modes. So pick and place, where we use the FluidFM probe to pick, to grab and relocate single cells or other microsite objects. The second FluidFM mode is injection and sampling, where we use a nanosyringe to penetrate into cells, and then by applying a positive pressure via the microchannel, we can deliver component to the cells. Or at the opposite, if we apply a negative pressure and under pressure, we can extract the content of a single cell. And the third FluidFM mode is what we call localized dispensing, which is dispensing some liquids in air or in immersion. We will see later, for example, for micropatterning application or a stimulation of neurons. From these three FluidFM modes, we can combine them or use some of them and apply them in a very broad range in life sciences, from gene editing. For example, if you inject CRISPR-Cas complex into cells and combining with the pick and place, you can even create new cell lines. Mechanobiology, you can measure forces, adhesion forces, thanks to the laser force feedback. Nanoprinting, single cell omics, virology application. But today, we will mainly focus on the application of this FluidFM mode in neuroscience. We have developed a different system at Cytosurge. The FluidFM add-on for people who already, for example, possess an AFM system. You can combine it. It's really powerful and affordable. But more recently, I mean, already a few years back, we have developed a standalone system, what we call the FluidFM Omnium, which really unleashed, I would say, the possibilities that are offered by FluidFM. And which is highly automated, as we will see later on during this this presentation. Here is a picture and video of the FluidFM Omnium, just to show you the ease of use. So we see this probe here that you can easily fill with 1 microliter, and this is the only, I would say, manual step. Then you place the probe into the system here in the XY stage that you can see also on the right picture here, and then everything is controlled via our software that we have developed that control both the X, Y, and Z stage of the FluidFM system, but also the Olympus IX83 microscope, which is an fluorescent microscope on top of which the FluidFM Omnium is set. This really allows to control via our software all the aspects of the microscopy, and then therefore allow us to perform long-term observation when you perform a FluidFM experiment of your cells. You don't see on this picture, but there are also a custom-made incubators that control temperature and CO2 level to really be able to maintain the cells. And it's especially true when you work with neurons. It's very important to maintain them in the right condition. It's a technology which is already quite well established with more than 100 installations worldwide, both in academic and industry, and which has been already published in more than 90 publications. We will see a few results from this publication during this presentation, some in very high impact factor like Cells or Nature. Yeah, I will give you an overview of a few of this publication later on. So let me now dive into FluidFM and how we can apply this technology in neuroscience. So I already present you with slides of the different FluidFM modes, and we will see that all of these modes can be particularly interesting when we work in neuroscience, in the neuroscience field. From the pick and place to really select neurons and isolate them, to organize them, for example, in a specific pattern, injection and sampling to transfect them, or to inject any sort of molecule into neurons that can be relevant biologically speaking, to the localized dispensing, we will see, to help the micropatterning and the creation of neuronal network. And talking about that, I would like to, so this is the broad range of applications that are enabled by FluidFM. One of the most important that we will talk is the ability, let's say the possibilities that it offers in building customized neuronal network. We will see also how we can easily stimulate neurons or also inject any type of compound into neurons for transfection, for example, but not only. And we can, as already mentioned, as it is the FluidFM system is placed under an incubator and control an epifluorescent microscope. You can also perform some long-time observation of your experiment and analyze some data. So the first application I'd like to discuss with you is about the creation of custom-made neuronal networks. And here it is enabled by the combination of two FluidFM modes, the pick and place and the liquid dispensing, that really allows on one side to really select the neurons, pick it and place it at the location that you want. And we will see with the liquid dispensing, you can also control the connection between neurons. We will see that in later slides. But first, let me explain you the principle of the pick and place with FluidFM. So we use, if you look on the right side of the picture, the FluidFM micropipette, which is a flat tip, to approach cells, apply a negative pressure to pick it, and then you can move the probe really easily to place it, for example, in another well or at any location where you want to place it. Here it's shown with adherent cells. It works both on suspension and adherent cells. For adherent cells, there is a few upstream steps that you need to apply. You can fill the probe with trypsin here. When you approach the cells, you apply a positive pressure to shower the cells, deliver the trypsin, which allows the cell to detach and then to be picked by the micropipette. This pick and place, FluidFM pick and place, has been already automatized. As you can see on the picture, you can select a lot of different cells, and then the system, thanks to the laser force feedback, will know when the probe has touched the cells, then knows what pressure to apply, will pick it, go to the location that you have predefined, and bring the cells there and drop it by applying a gentle positive pressure. So you see that on the video here, where it is happening. And here are a few pictures after the isolation. It was done here with CHO cells, where you see that all the cells survived after 24 hours and 48 hours. So the process is very gentle. So also thanks to this force feedback that we have, not only it allows you to automatize the system, but also to be very gentle because you can control the force that will be applied on each single cells. This is also working with neurons, as you can see on this video. Let me start the video here. It's a culture of mouse primary neurons that were produced from mouse brain. And before letting them grow and develop the neurons, you can easily also, like we did with CHO cells, select several of them and then select a few parameters for the isolation. The probe will approach. Here we approach a bit slower to the neurons because it's a more sensitive cells. We pick it by applying a negative pressure. Here the probe retracts. We can see it comes with the cells. And then it will move to another well where we have already placed a few cells here, and we will drop the cells by applying a positive pressure. What is interesting is, like with the CHO cells, the process is extremely gentle, and we can see on this picture where 9 neurons were isolated. So not all of them are viable after the isolation, but this is normal because from the suspension culture of primary neurons, they are already not all viable. And actually, if you compare the viability of the isolated neurons compared to a non-isolated control, you have similar rate of viability with 97% of viability in the isolated neurons compared to the non-isolated control. So the process is extremely gentle, even with very sensitive cells such as neurons. So what is interesting here is that you can already imagine that with this FluidFM technology, you could, for example, mix different cell types. If you want, for example, to study how neurons interact with, I don't know, a muscle cells, there's a neuromuscular junction. You could place a neurons next to isolated muscle fiber or other type of neurons. So here we have seen how with FluidFM we can easily pick and place cells, and this is the first step when you want to create neuronal networks. But what will be really interesting is if you could also control how these cells, let me reuse the laser pointer, sorry, how these cells will connect to the next ones that you have placed here or here. So how will you be able to drive the connection between the neurons? And here, actually, FluidFM can offer some solution by using the liquid dispensing mode. Here, where you can print, here I present you the mode where you can print any sort of substance on glasses. Here, it's just to demonstrate the precision that we can achieve. Here with DAPI on glass substrates with lines in the sub-micrometer range, and you can print any type of substance on any type of substrate. Of course, it depends on the chemical properties of what you are printing. When we use, for example, the FluidFM nanopipette for that, or we can also use a micropipette for larger structures. What is interesting is it has already been used in neuroscience, this, let's say, patterning ability to connect to control neurite growth. Here in this experiment by Dermont and its colleague, they have used a surface that was coated with PLL-JPEG, which is a repellent surface, here in yellow, and they have put some PLL, which is an attractive substance, to seed some microbrain, some group of neurons on top of this PLL structure. And with FluidFM, they have been able to connect these two microbrain structure by drawing a line of PLL to control, we can see it better on this picture, the growth of the axon between these two different structures. So you can easily imagine at a single cell level by combining the pick and place, what I have shown you in the previous slides, and this PLL coating, this patterning ability of FluidFM, how you could customize in very unprecedented ways your neuronal networks. What is even more interesting is that with this FluidFM liquid dispensing, you can not only micro-pattern, so create some structure on glass surface where the neuron will grow, but you can also stimulate neurons. If you fill the micropipette with a neurotransmitter, for example, like a glutamate, you can easily approach and control, place your probe on top of a neurons and deliver this glutamate on top of a specific neurons. If you combine, here it was performed by Ebersol and its colleague, where they combined a FluidFM add-on system with an MEA plate, and they could approach like that with FluidFM, deliver some glutamate on top of neurons and record at a single cell level with the MEA, also with calcium imaging, the reaction of the neurons to the glutamate delivery. What is very interesting compared to traditional method in the field is that you don't need to develop some caging strategy. I don't know if you are familiar with caging strategy, but normally what people do is that they put some glutamate on top of the cells. But because the glutamate is everywhere, if they want to control which cell is activated, you need to chemically modify the glutamate, and then you can activate it with light. It's called uncaging. This very cumbersome and time-consuming process is not needed with FluidFM because you can deliver directly, very locally, the glutamate to the single neurons that you want to activate, to stimulate with it. So the second application I'd like to discuss with you in neuroscience is the injection into single neurons. Injection of into cytoplasm or nucleus that you can control actually with FluidFM, which is very gentle. We will see injection of any sort of compound, plasmid, CRISPR, or any molecules that could be relevant to neurons, biologically speaking, relevant to neurons or neurodegenerative disease. And we will see also that when we perform injection with FluidFM, you can also measure the volumes that you have injected in each cell, which allows to draw some drug response curves, for example. So the principle of the intracellular injection is we use a nanosyringe. So this probe with a pyramidal tip here, very sharp, with an opening on the side, with an aperture on the side of the pyramid. And when the nanosyringe approach the cell, the laser force feedback will tell to the system that it has penetrated the cells. We can apply the pressure and deliver the payload into the cells, here directly into the nucleus. So this nano injection into cells was developed originally by Oran Guillaume-Gentil at ETH already a few years back, here on HeLa cells. You can see on the left force curve, so the force measurement of the approach of the nanosyringe into the cell. You have on the x-axis, the distance between the probe and the sample, and on the y-axis you have the force which is measured. And you can see in blue the approach phase, and in gray the retraction phase. You can see when you approach, when you start to touch the cell, the force starts to increase until there is this little bump, which is actually the sign that you have penetrated through the cytoplasmic membrane. And if you continue to approach, to apply some forces, you see a second little bump, which is the sign that you have perfored through the nuclear envelope. And then you can continue until you reach maximum forces, which is actually when you touch the substrate below, so the plastic substrate below the cell. She could show that you can easily inject and measure different volumes that are injected here into HeLa cells with small fluorophore here. But actually, we have already been able to show that any sort of compounds can be injected with this approach, from small fluorophore, here Lucifer yellow, to proteins like UHRF1 here, or even big protein like antibodies, small RNAs, CRISPR complexes, plasmids as well. Here, 2 plasmids that were co-injected together into CHO cells, expressing for one a nuclear GFP protein, and the other one cytoplasmic protein tagged with M-cherry. You can see on the top right picture here that you can really control injection in the cytoplasm. Here, for example, in this mouse hepatocyte. So these cells have 2 nuclei. That's totally normal because they don't finish their division. But you can also inject only into the nucleus, like here, for example, for this nuclear protein that was injected directly into the nucleus. Also of, no, here it was into FG2 cells. The approach is very universal and has been used already on a lot of different cell types, from stem cells to suspension cells like CHO cells, GERCAT, and mouse primary hepatocyte, as I already mentioned, HEC cells, but also neurons, like, for example, here, pig neurons, where Lucifer yellow was injected. It is extremely gentle, and thanks to the force feedback control, we can really reach high variability. The process itself of penetrating through the cells does not kill the cell at all. Of course, then depending on what you deliver to the cell, what amount you deliver, you might induce some toxicity, but the process itself of penetration of the FluidFM probe into the cell is extremely gentle, and really allows to work with hard to transfect cells like neurons, but also sensitive cells such like mouse embryonic stem cells. Here on the left, some transfection experiments that were performed at in Guangzhou in China at the Jinan University, where GFP plasmid were injected into neurons, or here on the right, some mouse embryonic stem cells that were injected with RNases in a nice publication in Nature that was published by a group working in PKU in Beijing. Here, a little video. Let me maybe, oops, sorry, let me go back to the video. A little video of an injection to show you how it works, how it looks like. So it's a view of our software here, and it's a video of injection into these cells that are Jurkat cells, so suspension cells. So here's the trick with suspension cells is to coat the plate with PLL, so it immobilizes a bit the cells when we will approach with our nano syringe here from the top into the cells, so they don't move away. We will see when I will start the video, but I want first to show you a bit the software, how it looks like, how easy it is to handle. But you have on the left some nice tool that can control the pressure, the position of the probe, and on the right, here we are already in the injection workflow, but you can have different workflow for depending on which application you want to do. Here for the injection, it's very simple, very straightforward. The first step is to manually select which cell you want to inject. So here you go and just click on which cells and where you want to inject them. Then you select a few parameters here. The set point, which is the force that will be applied. So depending on the sensitivity of the cells, you might play a bit on it, on this parameter. The approach speed, the retraction speed, but also the pressure that you will apply and for how long you will apply this pressure. And the retraction distance is how much the probe has to retract from the cell before going to the next cell to avoid that it scratch the cells when it moves from one cell to the other one. And then as soon as you will click on OK here, and I will restart the video, the process will be fully automatized. The cells will go, oh, sorry for that. The cell will go from one cell to the next one. We'll start to apply the pressure, and when it has applied the pressure for the time you have a setup, we'll move to the next step, next cell, and so on and so on. So here you can see live in real time, injecting into the first cell, moving already to the second cells, and then it will move to the third cell. And we will check here, it was Lucifer yellow that is injecting into this Jurkat cell. We try to always use a fluorescent marker to really monitor the success rate of the injection. So here we can easily switch to fluorescence mode with the software, and like that observe that the cells were successfully injected. You have to retract the probe because it brings a bit of background, as the reservoir is also filled with a lot of this Lucifer yellow. So I already quickly mentioned, but one of the advantages of injection with FluidFM is you can also measure the volumes that you deliver into each single cell. And this is allowed by the fact that we know exactly the dimension of the probes that are made of glass and therefore that are transparent. And we use then the fluorescence intensity of the probe to compare it to the intensity of the cells, and like that we can measure the volume that is delivered. So you can see here it has been integrated into our software. So you use the probe here as a reference and just give to the software the delimitation of each cells, and like that you can measure how many femtoliters were injected into each of the single cells. That can be particularly relevant when you are studying some drug response at a single cell level. So now, how can we apply this injection into neurons? And it has already been done by a few of our users around the world. Here, an example in China, where they injected some markers and observed their spread along the axon. It was performed here into C57 black 6 mouse hippocampal neurons. Here, and you can see they used the red dextran marker here to really monitor the injection into the cells, and you can really nicely observe its propagation through the axons. One of the most interesting application in using injection into neurons is, I already quickly mentioned it, transfection of neurons in culture. They are usually very difficult to transfect, these cells, because liposuction or electroporation are very inefficient or very toxic to neurons. You can usually not transfect plasmid with liposuction because as the cells never divide, it will never reach actually the nucleus where the plasmid needs to be transcribed to then express its payload. Here with FluidFM, you deliver directly to the nucleus, so it's not a problem, and you are very gentle, so you can easily transfect any type of neurons. They stay viable and they express here, for example, GFP from a very simple plasmid. Was done here in Guangzhou, and you can see the GFP expression 24 hours after the injection. The neurons is still viable and expressing the GFP. Another application I wanted to discuss with you today is what we call the sampling, extraction of the content of individual cells directly in their native environment. It has so far never been done with neurons, but I'm convinced there is a lot of interest in the field, and it could really be applied here in neurons to solve a lot of questions here. So basically with FluidFM, you can, like we did for the injection, penetrate into a cell, but instead of applying a positive pressure, you can apply a negative pressure to perform some cytoplasmic or nuclear biopsy from the single cell. What is extremely innovative and novel here with this technology is that it is non-destructive. So you can do several time point molecular analysis on the same single cell. So the principle was published and developed by Orhan Guillaume-Gentil at ETH in 2016 and published in Cell paper. So it's, as I already mentioned, very similar to the injection principle. So you target a cell with the system approach with force feedback control, which allows to be very gentle. Once you are into the cell, you can apply a negative pressure to extract the content, retract the probe, and then bring it to another substrate where you will perform the analysis. He originally performed some electron microscopy imaging of the extract, biochemical assay, but also some transcript analysis by RT-qPCR, and could show that really you could measure some expression from a volume that was much less than a single cell volume. So she shows that you could selectively extract from the nucleus or the cytoplasmic, and that the cell remain viable. So sorry, I'm not sure if we see the video here. Yes. So here you see it on video. Here, I don't know if you can see it very well, but the cantilever, the probe here is filled with oil. So actually, when you extract, you can see the interface between the cytoplasm or the nucleus that you extract and the oil here, if you look really attentively. But of course, you can also see that with fluorescence marker if your cell is expressing some fluorescence, and here it's much easier to see that you extract some content from these GFP cells here. It was also a paper published by Orhan Guillaume-Gentil, where this time she could show that from a single cell, she could even perform some mass spectroscopy analysis with MALDI-TOF from a single cell, still keeping the cell alive. It was very interesting to notice in this paper that even if you extract more than 60–70% of the cytoplasm with HeLa cells, at least, they still survive to the process. More recently, this year, the group of Bart Desplanques at EPFL in Lausanne developed a new application using this FluidFM sampling methods that he called LiveSeq to perform non-destructive transcriptomic profiling of an individual cell. It was performed in very similar approach. Before performing the extraction, they were first preloading the probe, the nanosyringe, with some RNA extraction buffer by approaching the buffer, applying negative pressure to load it into the front part of the nanosyringe before going to the cell, extracting the cytoplasm, and then depositing on another substrate to analyze the transcriptome. It's absolutely a huge breakthrough in the field, in the sense that it was the first time that they were able to show that it is possible to perform a transcriptomic analysis with less than a single cell, that this transcriptome is actually, representative of the transcriptome of the full cell. They could compare, and it's very similar. And also, the extraction procedure does not affect the transcriptome of the cell. So if you perform several extractions from the same cells, after a few hours, almost all of the gene will be expressed in the same way, except a few genes that are involved in the cell cycle, which is expected. So that was the last, let's say, application I wanted to show about FluidFM in neurosciences. And maybe just as a summary, I could present you that we have seen today that FluidFM allows 3 different modes: pick and place, injection and sampling, and localized dispensing, that all can be used or combined in neuroscience field to create customized neuronal network, to stimulate neurons and later observe them by delivering some neurotransmitter, for example, on top of cells. They can be used to inject into neurons. We have seen that for transfection, but you can imagine that also proteins can be injected, like protein involved in neurodegenerative disease or other biological aspect of cells. And it has never been done yet on neurons, but we are very convinced that the extraction from neurons is possible and can answer a lot of questions in the field. So with that, I would like to give you some time for the Q&A now. If you have some question to me, and hope you enjoyed this presentation.
Yeah, thank you so much, Paul. That was very impressive. Thank you. And I think to most of the participants in this session, the technology is quite new. So especially in neuroscience. So maybe as a first question from my side, what are the typical application outside of neuroscience that the FluidFM technology is used in industry and academia right now?
So one of the big applications that is really high, it's in the CRISPR field. So a lot of people use the nano injection approach to deliver CRISPR in hard to transfect cells. So it's true with neurons. It's true with a lot of different other cells that are known to be hard to transfect. And combining with the pick and place, you can even create some new cell lines in a very novel approach, which is really fast compared to traditional method, but also very precise, because you can inject a specific cells. With FluidFM, we couldn't see that in this presentation, but we record with our system the position of any event, so of every cell that have been injected, and you can easily on the next day come back to the same position, pick the cells, the real cells that you have injected, and isolate it. So in cell line development, to get monoclonal cell line, it's really speed up the process. That's one of the main application.
Mm-hmm. All right. So first question from the audience is, how does the probe recognize the nucleus of the cell?
So usually I mean, you can measure with the force feedback the penetration from through the cytoplasmic membrane and then through the nuclear envelope. But the way the nanosyringe is built with the opening on the side of the probe, I mean, everything has been really well studied, like where is the opening located, etc. And actually, if you target just in XY, if you are on top of the nucleus, you will always be, if you approach with a high speed, always be in the nucleus. And if you in XY control that you target on top of the cytoplasm, be in the cytoplasm.
Okay. Yeah, so one question that we often get is the question of throughput. So how many cells could you inject per hour or per day?
So that's an excellent question. So it really depends on the cell type and on what compound you inject. Some compounds are a bit more viscous, so a bit more difficult to inject. You need to spend a bit more time per cell. But I will say it's between roughly 200 and 500 cells per hour. Yeah, depending on the cell type and the compound.
Yeah. All right. So next question is, can this be applied to iPS-derived neurons too?
So it depends what application, but I guess every application, yes, can be applied. We know already. I cannot always show everything because some of our users, have some confidential information, but it has been done. The pick and place, at least, for sure, with iPS-derived neurons. And it was the same, like viability-wise, it was similar, the viability between the isolated and the non-isolated one.
Mm-hmm.
Yes.
And then we have a next question. Where do you see applications for FluidFM in neuroscience, for example, related to drug development or disease studies?
I think both. I mean, we are in contact with, I cannot, that's the same. I cannot divulgate all the information. We are sometimes in contact with some pharma company, and they're usually quite strict on their policy. But what can I say without? Yes, definitely in both. In drug discovery, but not only in neuroscience, but in many fields with hepatocytes and stuff like that. It's also very important here. But in neuroscience as well, and definitely in disease modeling, because you can easily edit the genome of a neurons, so you can create new disease model here. Yeah.
Okay, good. Then next question from the chat window. What experiments were done to ensure that the pressure from the probe does not disrupt the membrane structure and subsequently induce certain pathways, for example, disrupt tight junctions?
Okay, so I'm not 100% sure. So the tight junction, I have no data on that. I know, for example, the gap junctions, they are not affected. And it's clear when we use lucifer yellow for injection, because when you have gap junction in between the cell, lucifer yellow pass from one cell to the other. And we see that very clearly, for example, with CHO cells. When we inject lucifer yellow in the CHO cells, or with stem cells, it's even more obvious actually with embryonic stem cells. When you inject one embryonic stem cells, if you use lucifer yellow as a marker, the whole colony is green after the injection. The whole clone is green. So the junction are still here. It takes some time until it goes to all the cells, but they are still here, definitely. The tight junction, I'm not 100% sure, but the pressure that we applied is very, very precisely controlled to less than a millibar, with less than a millibar precision. So you can really adapt it. We really adapt it to each cell type. Like on the video I showed, it was on suspension cell in jerkat. It was 5 millibar pressure, for example. With CHO cell, we use 40 millibar. If we use 40 millibar with a suspension cell, jerkat, for example, you blow up the cells. But we can really control that. So you would have to optimize for each cell type what parameters have to be used. But we already have a lot of expertise in that to also give some insight to any of our users that would like to start with a new cell type.
Okay, and then we have another question regarding precision. Can you target single axons with FluidFM?
I think not. We never tried, but I think it's we are at the limit of the precision. The size of the aperture on the FluidFM probe is roughly 600 nanometer, but then the size of the pyramid, etc., it might be very tricky. It might be possible at the base of the axon. I'm not even sure, but we never really tried that so far. At least I'm not aware if I don't know everything that our user might have done, but I'm not aware of anything like that.
Mm-hmm. Okay,
that would be interesting, but yeah. Definitely.
So I think that's in general true that there are a lot of interesting potential applications. So maybe a question from my side. Do you perform proof of principle studies in your lab? So if somebody comes and with a new question like the axon question, would you be able to test that in your biolab in Zurich?
The short answer is yes. The longer answer, it's of course depend on the scope of the project, because of course our resources here in Zurich are limited. We don't have a full biology lab, etc. But of course we are always happy. We do that from time to time, and we're always happy to do that indeed. Mm-hmm. With neurons, it might be tricky because we don't have the full. That's one of the main issue with neurons, that we don't really have access to neurons here. With primary neurons, you need to have a fresh arrival all the time. You cannot keep them forever. So that was limit a bit this proof of principle here. But we have done a few, like the isolation video of neurons that you have seen was done here in our biolab. Yes.
Yeah. Okay. Another neuron related question. A key challenge with neurons is often that they come with low viability and die. Mm-hmm. Can FluidFM help research here?
Yeah, this is an excellent remark that we have faced as well, is that actually when you have this suspension of neurons, where you have seen where I was isolated the neurons, well, no, I was not, sorry, it was Maria, our other application scientist that did that, but where we were isolating the neurons, yes, maybe half of them are viable. I mean, it depends how well you prepared your neurons. You have to imagine that the neurons we had here, the preparation, it was coming from another lab. So with the transport, etc., we had maybe 50% viable cells and 50% already dead cells. In the where it can help, it's because we select which neurons we pick up. So already some that looks dead, we avoid to pick them. Does not guarantee 100% viability at the end. And we have seen on the picture of the 9 neurons that are isolated, 2 were dead. That we could not avoid, but we could optimize that. We could optimize with some staining, maybe some live dead staining, because we have this fluorescent microscope, and we could only select the one that are viable. So that's definitely a way we are exploring at the moment in this field. Yes, okay. Very, very, very good remark. Yes.
Yeah. All right. So if you have any further questions, please put them in the Q&A window. Otherwise, so what me in general excites with this technology is that finally robotics and automation has reached the single cell and even the subcellular level. Yeah, that we can manipulate cells, inject liquids like CRISPR-Cas and even extract fluids from inside the cell and analyze it, as you have shown in the papers at the end of your talk. So we are reaching further down in scale with robotics and automation, and I think that's a huge potential for different applications, not only for neuroscience. And I just see another question, which is, it looks very small. Do you have problems with clogging during the experiments? Also very good question.
I'd say it depends how well you prepared your sample that you want to inject. We use some specific coating on the probe. I didn't mention that, but when we do injection, for example, we have a very specific coating. It's called paramount, produced by a company in Switzerland here. As well, which avoid this clogging. If you do the injection without cutting the probe, yes, after 20-30 cells injected, you will clog the probe and you will need to unclog it, etc. But without cutting, you can inject several hundreds of cells, thousands. We have done sometimes. We don't know the upper limit really. Might depend on the compound. With some proteins as well, they might aggregate. So you need to prepare, maybe do some work on the aggregation of these proteins to avoid that, with then using different buffer, adapting your buffer that will mitigate this risk of clogging. But visually, I mean, for plasmid, for CRISPR injection, it works very nicely without extensive clogging, definitely.
Okay. Another question regarding viability of cells. What is the viability of the hard-to-transfect primary cells after intracellular injection? Do you notice an activation of the interferon pathway?
we never checked the interferon pathway. That I cannot tell you. I know they are viable. For example, the primary hepatocytes that I've injected, the process itself of injection does not kill them at all. The process of penetration with a FluidFM probe, etc. I mean, we had very high, more than 95% viability. After that, I was, let me remember, I did injection only with small RNAs or stuff like that. So with that, at least it was definitely fine. Of course, if you want to inject some drugs, some stuff, the concentration might have an impact on the viability.
Okay, and then another neuroscience-related question. What about typical neurotransmitters? Any special preparation there?
Yes, so that's what I tried to briefly mention. This is one of the advantage. We don't need to any specific preparation. Here in the publication I shown, it was done with glutamate, but very simple glutamate. You don't need to prepare it at all. You don't need to cage it like it is done normally in the field, because you will really deliver it directly on top of a single cell and not to the neighboring one. You might have some diffusion, but you can control the pressure to limit the diffusion to the very specific cells that you want to activate.
Okay, thank you very much. All right, so we're coming close to 5 p.m. Thank you very much, Paul, for introducing us into the neuroscience applications of FluidFM and into the technology itself. It was again very interesting, very exciting, and also thanks for answering all the questions. And to the audience, thank you very much for joining for our today's webinar. And if you have any further questions, please do not hesitate to send us an email. We'll be happy to answer this. And if you have any idea what you would like to test, please also ask us and ask Cytosearch. We may be happy to answer the questions with a test that could be performed in the biolab in Zurich. And of course, there will be a recording available that you can, if you like, forward to interested colleagues. Please let us know if you're interested to get the recording from this session. All right. So thank you very much, everybody, for joining. We will have our next webinar in September after the summer holidays, in September 21st, with PantaBio and a software solution to connect laboratory instruments to each other with a very simple and smart solution. It's called the glue layer solution from PantaBio. So it will be the next webinar on September 21st. We would be happy if you join, and let's stay in contact. Thank you very much for today, and enjoy the rest of your day. Bye-bye.
Thanks, everyone. Bye-bye.